Thought we probably need to get started so we can kind of keep on time. I'm already 15 minutes over the time schedule, but what I want to do is just a couple things to update you on. First of all, I do want to make sure that we understand that a lot of the work that was done behind this was done by the Caslin Group, and both Debbie Sapperoni, Robin Quinter, and Ann Conlon are the people here that are making this symposium work. And Debbie's been extraordinarily good at this for the last couple of years. These don't happen without people behind the scenes getting this done. And so I'd like a little hand of applause for that group. So please. Second thing, I want to again thank the sponsors. And for you, communications, you will get a communications, a survey, and please send that in to give us insights into how the conference went for you. I think that what Debbie told me is that you send all that in, you can get whatever slides they have that are permissible to give out from this talk. So if you wanted some PowerPoint slides that are available, you can get them through that thing responding to the survey. Remember this afternoon, the posters will be over here, and please spend some time going through those. Really had a pretty good number of posters, and particularly for the young people and stuff like that, it's always nice to go by, look at them, and for all the efforts that they've done. There will be an awards presentation for best posters at about 4, I believe, down here, about 4:45. There it is. And I'm not sure, Elon, whether we also give the award for the Twitter thing at that time too. I suspect we do. I wasn't informed exactly when that was going to happen, but somebody out there is trying to win $250, I suspect. And then, of course, tonight we will have the dinner that starts at 6 o'clock. Excuse me. With that dinner, hopefully that's right. 6 o'clock. I will check that. The dinner, I think it says 6 o'clock here, but I had it someplace out a little bit. But the important thing is, actually, there was somebody talking on the ECHO program, the Indian ECHO program, and so it's one of the things we've done at these meetings is have somebody locally come and talk about issues around the environment and the geography and stuff. And so please be ready for that and try to attend. Okay, to get us started this morning, again, as you've seen with this program, there's a lot of eclectic type things. We go on different areas. Yesterday, talked a little bit about host, about education and stuff. And today, actually, this section is the organism. And I think you should be hopefully pleased. We have three talks on this, one from the crypto community, one on C. aureus, and the other on Talaromyces. All these speakers are outstanding. It's your ability to see the thought processes around these organisms. And it's my pleasure to start off this morning with Kirsten Nielsen. She's a colleague for many years and very kindly as a PhD researcher, basic science researcher in Cryptococcus. When I asked her to do this talk on genotypes, which is kind of dear to my heart for sure, she said yes, no question. And so she's come to do this on cryptococcal genotypes. Do they mean anything to the clinician? She didn't make that topic up. I did. So she can do whatever she wants with it, but I appreciate her coming to do this. And for the first speaker, Kirsten.
All right, so as John mentioned, he gave me the very controversial task of trying to convince you that you should care as clinicians about what genotype your patients with cryptococcosis are infected with. And so today I'm actually going to talk exclusively about Cryptococcus neoformans infections, because I think these are really going to be the hallmark infections that you're interested in. And that is because these infections are predominantly immunocompromised individuals. And so this raises a fascinating question of Is it simply that the patient is susceptible to this infection, and so they're going to get that infection no matter what, and the outcome of that infection is going to be the same no matter what? Or is it actually that even in the context of immunocompromise, differences in the strains that are infecting these patients may actually influence how that patient is responding to that infection and ultimately what the clinical outcome is going to be? And so we first started answering this question, we and others in the early 2000s, by first asking, do we see differences in the major clades of Cryptococcus and patient outcomes? And so in Cryptococcus, we have 3 major clades, VN1, VN2, and VNB. And then in each of these, we have clades that we refer to as sequence types, okay? So in Uganda, we showed that the most common clade that we're seeing is sequence type 93. It accounts for about 60% of the infections in Uganda. And we were able to show that compared to the other sequence types that are present in Uganda, ST93 isolates have higher mortality rates in patients. And working in an ex vivo cytokine release assay, we were able to show that an ST93 genotype did in fact produce a different immune response in healthy individuals compared to another sequence type. And these studies have really now been done throughout the globe in different patient populations. And the trends are still holding. So ST93, for example, is not a common sequence type that they see in South Africa, yet they are seeing similar disease characteristics with some sequence types showing more mortality than others. So if we look within the sequence type, we also see quite a bit of diversity, and this is rather interesting. So here I'm just plotting patient survival versus sequence type, and each one of those little dots indicates a patient. Okay, so you can see that in some sequence types like ST93 or ST71 on the left, that, or maybe on the right for you guys, nope, on the left, that many of the patients with these sequence types are able to survive those infections and do quite well. In contrast, if you look on the far right, you can see ST40. All of the patients with that sequence type succumbed to those infections. But what I think is perhaps more interesting is all the sequence types in between. And I suspect if we did a deeper dive, we'd see that this is going to be true for all sequence types. And that with these infections, there's a population of patients that live and a population of patients that die, right? And this really led us to 2 hypotheses. The first one is, as John likes to say, the host is the most. And it is differences in the host that is really determining the outcome of those infections, right? But there is another hypothesis, and that hypothesis is that it's differences in the pathogen and how that pathogen is interacting with the host that will change that clinical outcome. And so how do we address this question? Well, so we decided to actually change the host, right? So we took these same strains that were infecting these same patients, and we used them in a mouse model of cryptococcosis, and this model uses the AJ mouse background. And so what we found is that in patients that the isolate from the patient killed the patient rather quickly, we saw that it also killed the mice rather quickly. Isolates from patients that survived tended to have longer survival in the mouse, if not complete survival in the mouse. And so this really showed us that those isolates were behaving the same way in both the human and the mouse. And this really told us that that the strain is having a very dramatic impact on that host, on that disease outcome. And really led us to say, okay, now that we know that that's the case, let's study it a little bit more. And so to study it, we said, okay, let's take ST93, one, because we have a lot of those strains in the laboratory, and two, also because you can see that it's got a very nice distribution between the strains where the patient survived and the strains that the patient succumbed to the infection. By looking within the same sequence type, we're actually limiting that genetic diversity in Cryptococcus, and that will allow us to actually perhaps dig down and see what the genetic polymorphisms are that are impacting patient outcome. To better understand the infection and the infection process, and because we know that the mouse model that we had was a good surrogate for human disease, we infected about 35 ST93 clinical isolates and another 15 isolates that were not ST93 in this mouse model. And instead of just looking at the survival of the mouse, we now looked in a little bit more detail at the course of the infection, what the immune response was in the lungs, what the CFUs were, to better understand how these strains were causing disease. And so, as expected, we saw that a lot of our strains, I'm just showing you 3 here as an example, had what we might classify as a typical virulence profile in the mouse. And so in all of the slides I'm going to be showing you, I'm going to show the survival of the mouse on the x-axis relative to KN99. So this is a large number of mouse experiments, and we couldn't do all of them at exactly the same time. My graduate student would have revolted. And so we did this across the course of 10 different experiments. And in each of those experiments, we included KN99 as a control so that we could normalize across those experiments and be able to compare virulence characteristics. And so that's why you might see a negative number. That's because that mouse succumbed faster than a KN99 reference mouse did. And so in these plots, you can see that all of the mice succumbed to the infection in a rather common survival profile. When we looked at the CFUs in the lungs, again, we saw pretty decent CFUs in the lungs. All of the clinical isolates are running a little bit lower in CFUs than the reference in the lungs, but they're not actually statistically significant. I forgot the statistical significance on this graph, but I think you can see where it is. What we did find is that even though the survival curves may have looked the same or very similar, we did have a subset of these mice with what we would classify as typical virulence that had very low CFUs in their brain. And what this suggested to us was that these isolates were causing cryptococcal pneumonia, and that was most likely what was killing these animals instead of cryptococcal meningitis. And so if we look at the data from all of the strains in ST93 that we've tested, you can see that we have a nice distribution of strains with typical CNS disease. Now you notice that the non-CNS disease is clustered at the bottom. And so in ST93, we actually have 2 subpopulations, and we found that these non-CNS diseases were all clustering in one of those subpopulations, which we call ST93B. So we also saw strains that produced latent infections, and we have developed a mouse model for latent infection that we recently published this year, and these strains are behaving in a very similar way to the one that we use in our latent mouse model. So you can see that very few of the mice succumb to the infection, yet when we look in their lungs, we see that while they don't have very high CFUs, they do have CFUs in the lungs, suggesting that the mouse is able to control this infection. The other important thing to note is that not all of the mice have clear brain CNS. And we saw this in our latent mouse model as well, where some of those mice we do see dissemination to the brain. We're currently determining whether this is actually a reactivation phenotype. as we saw with this non-CNS infections, these latent infections seem to be clustered predominantly in the ST93A population. We were also really, really surprised to identify a hypervirulent population. So we didn't see this in our original study that didn't use as many strains, but as we expanded the strains, we found a number that were hypervirulent. And so for those of you that work in Cryptococcus, and especially in mouse models, that you know that the CAN99 alpha strain is considered extremely virulent. These strains are even more virulent. So they're shifting those survival curves usually by about 5 days. These mice are dying very rapidly. When we look at their CFUs at time of death, we see that the lung CFUs are pretty similar to CAN99, maybe a little bit lower than we're seeing in the other clinical isolates. And the brain CFUs are also much lower. And so this suggested to us that wasn't an overwhelming fungal burden that was causing mortality in these mice, but instead perhaps it was how the mouse was responding to that infection. And once again, these hypervirulent isolates are associated with only one of the clades. And so this is giving us a little bit of a clue that perhaps the genetics of the strains are impacting disease and also how they're interacting with the host. And so if we look at all of these strains that we've done so far, you can see that we really see a continuum of disease, where we've got hypervirulence on one side, and we've got latent infections on the other side, and then we have quite a bit of variability in the middle. And so we wanted to know whether or not we could understand what the underlying reason for this variability in disease was. And so, like any good microbiologist, we went into the laboratory, looked at all the host mimicking conditions we possibly could in vitro in the laboratory, and we have not found a single condition that mimics the phenotypes that we're seeing and the trends that we're seeing in vivo. And so this does look like it's an in vivo phenomenon. So we've also looked at the host immune response to see if that can give us any clue as to what's going on. And as you might imagine, when we look at the latent infections, we're not really seeing a robust immune response in the lungs in terms of the cytokine profile that really can tell us what's happening. Now, as I mentioned, we have a latent mouse model now, and these strains are actually behaving very similar to our latent mouse model, where when we go in with cellular profiling using flow cytometry, we can see that these mice are inducing type 1 Th1 mediated immune response with a few caveats to that statement. In the typical virulence mice, we're seeing that they're predominantly producing a type 2 immune response with IL-4, IL-5, IL-13 levels quite high. And this also is consistent with what we know about CAN99 alpha, who is our reference strain for this virulence. And so this makes sense. But what we were really surprised to see is the immune response in the hypervirulent strains. So we anticipated that these hypervirulent strains would be eliciting a neutrophilic response, an IL-17 response, because we know that in mutants of Cryptococcus that tend to be hypervirulent, they tend to produce neutrophilic responses. Or we thought we might see this super active Th2 response that was simply more than what we saw in the other ones. And this is not, in fact, what we saw. So I'm showing you 3 of the strains here, but we've seen this for every single one of our hypervirulent strains, is that they actually have a mixed type 1, type 2 immune response. And in many of the strains, we're seeing a really dominant interferon gamma signal. Now, for those of you that know much about Cryptococcus, the hypothesis is that interferon gamma is a good thing, right? But in these particular infections, interferon gamma, when combined with even moderate Th2 or type 2 immune response, may actually be detrimental. And so I think it's something that we really need to that you need to consider moving forward. The other thing is, does the host matter, right? So all of this was done in a single mouse background, and that's really so that we could control the host. But it is important to note that the host background does make a difference, right? And so here's a really good example. It's all of the hypervirulent strains that are hypervirulent in the AJ mouse background. If we put them in the black 6 mouse background, you can see that this attenuates the hypervirulence and really shifts them away from being hypervirulent. And again, if we look at that immune response, you can see that the immune response is also fundamentally different in these mice. And so here I'm just comparing KN99 infections as a control in AJ black 6 and BALB/C mice, and then one of the hypervirulent strains, 247. And you can see that really strong interferon gamma response is specific to the AJ mice and is not seen in the black 6 and the BALB/C mice. And so this does suggest that how the host responds to that infection is likely also changing the clinical outcome. So what are the genetic differences? Can we drill down and find the single nucleotide polymorphisms or the modifications in the cryptococcal cell that impact that disease outcome? And so for these studies, we thought it was important to do them in humans. And so we've done that first. We are going back and doing a similar genome-wide association study using the mouse data that I just presented. But we've done it in humans, and I think for this audience, that's much more important to show. And so in collaboration with David Boulware and David Maya, we have been able to look for associations with genetic characteristics or with human clinical disease characteristics such as patient survival, both immune and microbial characteristics of the clinical disease. We've looked at the CNS human response in these patients, or the CNS cytokine response in these patients. And then we did throw in a few in vitro characteristics of these cells that we thought may be important. So how rapidly they grew in the laboratory, how much chitin they had, and then also in vitro assays looking at their ability to interact with macrophages. And so in this strain population, even though the ST93 isolates are all within the same clade, they had about 100,000 SNPs that were different between the strains. And so we focused our analysis on those SNPs that would be predicted to have a fitness effect, so that were in the coding region of the gene and would change the protein, or that were in regulatory regions of the gene that might change the expression. And so in doing that, we identified 7 genes that had variants that were associated with patient survival. And so I've got the genes listed here, as well as the number of variants, and then the phenotypes they were significant for in our genome-wide association study. And so what I want to highlight here is that while yes, all of them were linked to survival, we also get a lot more information by looking at a larger data set. So you can see in all cases, these genes were associated also with changes in the immune response that was observed in these patients. And in fact, we saw very few of the changes were actually associated with how well the organism was able to grow or survive within the host. And so this really highlights that the immune response is most likely a major driver of virulence. When we look at all changes focusing on immune response, you can see that now we can identify 40 different genes. Only one of the genes that we identified was actually previously linked to virulence. And so this really suggested to us that perhaps we have a really interesting phenomenon going on. So we've dug into these genes in a little bit more detail to figure out what they are, because many of them are unstudied. And we noted that 50% of the genes are cell surface associated. So they're either cell surface proteins themselves, excreted proteins, genes involved in cellular excretion, or genes involved in modification of components that are on the cell surface. And so this really suggests to us that what's likely going on is that the genetic differences between the strains are changing the antigens that they're presenting to that host and inducing a different immune response to that, from that host. Now, of course, how the host sees those antigens and how it responds to those antigens is going to be host specific. So in conclusion, the mouse model can be combined with clinical studies and perhaps should be combined with clinical studies to define factors that influence patient outcome. We know that we can identify genetic differences in Cryptococcus neoformans isolates that do impact human clinical outcome. And we also know that these impacts are really focused on how the host is responding to that pathogen. And so the host immune response is a major determinant of disease. So going back to John's question of, does genotype mean anything to you as clinicians? Well, hopefully I've convinced you today that yes, isolate genotype does influence clinical outcome and in fact can dramatically influence clinical outcome. And hopefully I've convinced you that genotype impacts the immune response, which we also know from many different studies, from many different individuals in the field, that immune response is critical. But can you do anything about it right now? And the answer to that question is no, right? So currently there are no immune modulatory therapies that you can use. Now the challenge is lots of people are working on immune modulatory therapies, right? But for an immune modulatory therapy to really be beneficial, you're going to need to know not only what immune response is being elicited in that patient, but also what immune response is that pathogen trying to drive in that patient. So you can figure out, how do I move that dial to the most beneficial aspect for disease? So with that, I'm going to stop. The graduate student in my lab who's done most of this work is Katrina Jackson, who is acting as solo in this figure. And I also want to point out that these studies could not have been possible without an absolutely fantastic interaction between David Boulware and David Maya, who run clinical trials and clinical data. And by combining expertise and the ability to do basic science with the power of these clinical trials, we can do so much more. And so I really recommend everybody that's setting up clinical trials to think about how you can use this data in unique ways, not just to ask the question of your clinical trial, but also other fundamental questions in the field. So thank you.
Kirsten, thank you very much. For you, just a little background. You see that KN99. That's our H99 strain that she made congenic and made it back and forth 10 generations. So KN stands for Kirsten Nielsen.